Welcome to Dr. Ravichandra official YouTube channel. In the last video, we have discussed necrosis. Necrosis is a spectrum of morphological changes largely resulting from progressive degradative action of the enzymes on a lethally injured cell. Cytoplasmic changes in a necrotic cell will be increased eosinophilia, cytoplasmic vacuolation, myelin figures, and calcification. Nuclear changes will be karyolysis, pycnosis, and karyorrhexis. Types of necrosis: coagulative necrosis caused by denaturation of intracellular proteins, example myocardial infarction. Liquefactive necrosis caused by enzymatic degradation of tissue, example brain abscess. Gaseous necrosis caused by both enzymatic degradation of tissues and denaturation of cellular proteins, example tuberculosis. Fat necrosis: traumatic fat necrosis is seen in breast and buttocks following a blunt trauma, and enzymatic fat necrosis is seen in acute pancreatitis. Now we will discuss apoptosis in detail in this video. Please like, share and subscribe Dr. Ravichandra official. Apoptosis is a pathway of cell death induced by a tightly regulated intracellular program in which cells destined to die activate the enzymes that degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. Apoptosis, that's why, is called as internal programmed cell death. It is a form of cell death designed to eliminate the unwanted host cells through activation of coordinated internally programmed series of events affected by a dedicated set of gene products. Apoptosis can be physiological or pathological. Apoptosis in physiological situations is seen in embryogenesis, hormone dependent involution in adults, cell deletion in proliferating populations, cell death by cytotoxic T cells, elimination of harmful self-reactive lymphocytes, death of the host cells that have served their useful purpose. Apoptosis in pathological conditions is seen in when there is injurious stimulus which is lethal to the cell, cell injury in viral diseases, pathological atrophy in parenchymal organs after the duct obstruction, and cell death in tumors. Injurious stimulus, when removed, if it reverts back, it is cellular adaptation, and if it is removed and cell go back to the normal, it also can be called as reversible cell injury. If the cell damage is not reverting back after removing the stimulus, then it is a irreversible cell injury. Necrosis and apoptosis are examples of irreversible cell injury. In necrosis, there may be a stage of reversible cell injury, but I doubt there is a reversible stage in apoptosis. Coming to morphology of apoptotic cell, there will be cell shrinkage, chromatin condensation, formation of cytoplasmic bleps, and apoptotic bodies. Phagocytosis of apoptotic cell by macrophages will be seen. In this picture, you will be able to appreciate a normal cell and a necrotic cell and a apoptotic cell. In necrosis, there will be enzymatic digestion and leakage of the cellular contents, whereas in apoptosis, there will be formation of apoptotic body and phagocytosis of the apoptotic cells and fragments. Coming to histological changes, single or cluster of cells are involved in apoptosis, and a apoptotic cell appear as an oval or round mass of intensely eosinophilic cytoplasm with dense nuclear chromatin fragments. There will be no inflammation in a apoptotic cell or process of apoptosis. There will be quick phagocytosis of the dead cells. This is a picture showing a apoptotic cell and apoptotic body. This is a one more picture of a case of viral hepatitis where there are numerous hepatotic bodies. Mechanism of are cellular events in a apoptosis. There will be cell signaling, control and integration, execution, and removal of dead cells. The signaling and control and integration will come under initiation phase. Execution and removal of dead cells will be in execution phase. Initiation phase is again divided into extrinsic pathway and intrinsic pathway. Extrinsic pathway is a death receptor pathway, whereas intrinsic pathway is also called as mitochondrial pathway. Signaling will be with the help of transmembrane signals, suppress the pre-existing death programs and initiate a death cascade. There is a receptor called as tumor necrosis factor receptor oligomerase leading to activation of the initiation caspases and a cascade of enzyme activation culminating in cell death. So this is death domain pathway or extrinsic pathway where a FAS ligand gets attached to FAS receptor forming multiple death domains FAS death domain. This will activate a procaspase 8 into caspase 8 which will further activate the execution caspases resulting in apoptosis. This pathway is called as extrinsic pathway or death domain receptor pathway. Control and integration. Direct transmission of the death signals by specific adapter proteins to the execution mechanism. Regulation of mitochondrial permeability by members of BCL2 family of proteins. There will be mitochondrial permeability transition and there is a apoptotic trigger called as cytochrome C which will bind to cytosolic proteins APAF1 pro-apoptotic protease activating factor and activates them triggering execution caspase activation. BCL2 suppresses the apoptosis by preventing increased mitochondrial permeability. The mitochondrial matrix cytochrome C is released into the cytosol and it will activate the procaspase 9 and results in formation of active caspase 9 which will further activate execution caspases thereby resulting in apoptosis. Other pro-apoptotic proteins like AAF, APF, AF1 will bind to and neutralize, thereby inhibiting the apoptosis and thereby showing impact on execution caspases and thereby apoptosis. Among the BCL2 family, BAX and BAD will promote apoptosis, whereas BCL11 will inhibit apoptosis. For easy remembrance, remember in BAX and BAD there is A, so it promotes apoptosis. In BCL11 there is Cl, that is close apoptosis. So it is a anti-apoptotic factor. Execution occurs in four steps: protein cleavage, DNA breakdown, protein cross-linking, and phagocytic recognition. Protein cleavage occurs with the help of caspases activation. Caspases cleave cellular proteins, lamins, leading to breakup of nuclear scaffold and cytoskeleton. Caspases activate DNase, which degrades the nuclear DNA. The initiation caspases, remember, in intrinsic pathway it is caspase 9, and in extrinsic pathway it is caspase 8 and 10. In both the processes, the execution caspases will be caspase 6 and 3. DNA breakdown into oligonucleosomes in multiples of 180 to 300 base pairs using calcium and magnesium dependent endonucleases. These DNA fragments are identified by a technique called as agarose gel electrophoresis. In apoptosis, there is regular fragmentation associated with activation of P53 gene. That's why we'll get a ladder pattern in agarose gel electrophoresis, whereas in necrosis you can appreciate a smeared pattern. So this is a picture of showing agarose gel electrophoresis. First one is normal, second one is apoptosis showing ladder pattern, and the third one is showing smearing pattern, that is necrosis. A, B, C. A is normal, B is apoptosis, and C is necrosis. Protein cross-linking via transglutaminase activation will convert soluble cytoplasmic proteins into covalently condensed shells, leading to formation of apoptotic bodies. Last step, phagocytic recognition. Apoptotic cells express a phosphatidyl serine in their outer layers of plasma membrane, flipped from the inner layers. Some will express thrombospondin, an adhesive glycoprotein. Other proteins secreted by the phagocytes may bind to apoptosis or apoptotic cells and opsonize the cells for phagocytosis. So this is a picture showing the entire apoptosis process. Regulators are BCL2 family members. BAX and BAD promote apoptosis, whereas BCL11 will oppose apoptosis. Cytochrome C is the trigger, and initiation caspases will be caspase 8, 9 and 10. Execution caspases will be caspase 3 and 6. The pathways will be intrinsic pathway, mitochondrial pathway, extrinsic pathway or death domain pathway comes under initiation phase, and in the execution phase there will be endonuclease activation and breakdown of cytoskeleton and P53 activation, resulting in formation of apoptotic bodies and later phagocytosis of the apoptotic bodies, leading to phagocytosis of dead cells. Clinical significance of apoptosis. Tumors have apoptotic index. It is a measure of proliferation. Apoptosis in tumors increases following irradiation and immune responses. Measurement of apoptosis in vivo or in vitro following treatment may predict effectiveness of the therapy. So with this, apoptosis completes. So what are the differences between a necrosis and apoptosis? Necrosis and apoptosis can be differentiated using 7 points. One is cell size. It is enlarged in necrosis and reduced in apoptosis. Nuclear changes will be karyolysis, pycnosis and karyorrhexis in necrosis, whereas nuclear fragmentation will be seen in apoptosis. Plasma membrane is disrupted in necrosis, intact in apoptosis. Cellular events or contents will be leaked out and there will be enzymatic digestion. Cellular contents are intact and they are released into apoptotic bodies. Inflammation is present in necrosis, whereas it is absent in apoptosis. Necrosis is always pathologic, whereas apoptosis can be physiologic or pathologic. Across gel electrophoresis pattern will be diffuse smearing pattern in necrosis, whereas it is ladder pattern in apoptosis. Thank you. Please like, share and subscribe Dr. Ravichandra official for more interesting videos.